Okay, 10.2.3 and 10.2.4 are going to be talking about this thing called gene linkage or linkage groups. And what this is saying is that when we did the dihybrid cross, if you remember, we were looking at two chromosomes, each with genes on them. So maybe we had like yellow and green and like smooth and rough. In this, we're only going to be looking at one chromosome, but that one chromosome has two genes on it. So let's draw one here. We'll use this example. So here's our one chromosome, a homologous pair, one from your mother and one from your father. And let's make them heterozygous at both loci. So that's the gene for C, whatever that is. I forget what we're looking at here. And also we have the allele for W. We have a big and a small. And very important, and this is kind of weird, when we're doing linkage groups, when we're talking about how they move, we write them sideways. So just think of like timber, and we knock this chromosome down so it's on its side. So that's what you see down here. These lines are just chromosomes that are laying down. So we have big C, little c, just like we had here. And then we also have the big W and the little w. And that is how we note, that is our notation when we're talking about linked genes, genes that are on the same chromosome. And because they're on the same chromosome, these alleles, little c, little w, are going to travel together, and that creates very different inheritance patterns. So let's practice one. So let's go to this slide. We're going to change the letters. And in this situation, we're looking at color. So the pea plant can be purple, which is big P, or it can be white, which is little p. And we're also going to be looking at the length of, I think it's a pollen, some part of the plant that either can be long, dominant, or little l for short. So remember, because there's one centromere here, this is considered one chromosome. So in this situation, we have an individual that's heterozygous at both loci. So just to make things simple, let's only look at one of these chromatids. So we're just looking at these two. So one is a big P and a big L, which we represent there, right? And the other one is a little p and a little l, which we've represented here. And we can do an inheritance cross, for example, a test cross. So let's do a test cross. If you remember, a test cross is taking an unknown heterozygous, an unknown dominant phenotype, and crossing it with a known homozygous recessive. So if it's known homozygous recessive, we know that each one of these chromosomes are going to have a little l at the locus, and also a little p at each loci. So that's how to note that. How would we draw it? We draw one of the chromosomes with the little p and the little l, and we draw the other chromosome with the exact same thing because they're homozygous recessive at both locus. And then we do the cross. So if we do the cross, I've got a box here on the next page. And remember, because they travel together, those alleles travel together, we must have here one big P, one big L, right? If you remember from our group, it was big P, big L. And on the other one, it was heterozygous at each loci, so there'd be little p, little l. So they have to travel together. So little p, little l. And it was a test cross that was homozygous recessive at both locus. So if we drew the chromosomes real quick, we have a little p and a little p, and also a little l and a little l, and they travel together. So this little p and this little l would come here, and this little p and this little l will come here. And then you just do the scenarios. So if this gamete met this gamete, it would be big P, big L, little l. If this gamete met that gamete, it would be little p, little l, and so on. And from this, you can determine the phenotypic ratios of your pea plants. Here we'd have a purple long, for example, and here we'd have a white